So um, I'm using the microphone on the computer today, so if it's not as good as the headset, please let me know by Slack. So the plan for today, well, I'm going to start to talk about pipes in R because pipes are used in the textbook and often online in examples, so you need to know how to read them at least. Sorry, just letting my dog in the house. Okay, so the second thing, main thing, is really to go back to the breast cancer transcriptome data set and talk a little bit about this whole cycle as a whole, how this exploration cycle works. And we'll touch on models. Arguably, the rest of the course, in some sense, is about different ways to model. And we have different ways to model because there's different types of bioinformatic or computational biology challenges. So it's a large topic that can't be covered in one lecture per se, but I'll try to give the context. We're going to focus a little bit more today on the gene expression side of the breast cancer transcriptome data set rather than the clinical pathological sample data. And in fact, this will complete part one of the textbook, and it completes conceptually part one of the course. So today's material inclusive marks the end of the line covered by the first midterm. So today's material could be tested on the midterm. But conceptually, it's really the end of describing in detail this box here. And we're certainly going to use these tools that we've been learning throughout parts 2 and 3 of the course, but this is really a milestone here because we have the basics now of data science. Part 2 is going to focus a little bit on this upstream region called data wrangling. That's the process of importing data from a database or a resource and bringing it into a Tibble. And in parts 2 and 3, really, we'll talk at various stages about communication. You're going to learn a little bit of R Markdown and R notebooks, etc. Okay, so let's begin. Okay, so pipes can actually simplify R code, but beginners don't typically think they do. They can be quite confusing. So if you have trouble understanding them, talk to the TA or I. It's one of those things where once you see it, it's sort of easy, but before you see it, it's just like, what? I don't get it at all. So just pipe up if you're not understanding. Okay. So pipes are part of the Magritter package, which was introduced in 2014 by Stefan Milton-Bach. I think it's playing on this René Magritte's famous painting about representation, etc. So it's part of the tidyverse, so when you load the tidyverse, you have access to it. As I mentioned, I don't usually prefer to teach pipes to beginner programmers, but because there's so many examples in textbooks and online, I think you need to at least know how to read them. Okay, so let's suppose that I wanted to compute a function that looked like this. Just an arbitrary function. It takes one value, parameter x, and first you take the tan h of x, then you want the exponent of that resultant value, and then you want to take the square root of that resultant value, and then you want to take the log of that. Well, the first approach that I might call a newbie approach is as follows. I set my variable x to be whatever I want. I set x1, a new variable, to be the tan h of x, and then I set x2 to be the exponent of the result from the previous guy, x1, and then again x2 comes in for the square root to form x3, and then my answer is the result applying the log to x3. There's absolutely nothing wrong with that answer. It's perfectly good. It's step by step, and I actually encourage new programmers to program in this way. It's clear and it's correct. Two great criteria. A slightly more, I don't want to say advanced, but succinct way that has some advantage is as follows. I basically assign my value x as before, but now I just use one variable. There's not really any reason to create x1, x2, x3. I can just keep using the same variable by assigning the answer back to the same variable at each step. So let me give you an example here. Okay, so um, I'll go over here. How does that work? I think it's informative to look at these things closely. So at the beginning, we create a variable called x, right? And there's, remember, three things happen. The box assign the name to it, and the value 5 is inserted in there. And the first step is to take the answer is set equal to the tan h. I'm just going to abbreviate here to this, of x, right? Now, because x is available in the environment, it knows to replace x by 5. And now the tan h of 5, I think this is, I'm not sure, but it should be very close to 1. You can check for yourself, but it should be something like this, okay? And now it creates a new variable, because it says assigns to this variable answer. And so now the three things occur here. New variable bound to this box that has this value 0.9 ... Now the next step is that we want answer to be the answer, the exp, the e to the, e to the answer, right? Okay, so answer exists in, now how does R evaluate this? Because I think some beginner programmers get confused that answer's on the left and right hand side. Let's just go slow and treat it the way R does. So R comes in and says, well, okay, answer, I'm going to assign everything to this variable answer. It exists. I already got a box for it. It's assigned equal to whatever the result of this expression is. Okay, so exp, well, I know what that is. That's the exponent function. I'm going to compute e to whatever's inside of here. And whatever's inside of here, answer, it exists in my environment. So I can replace this by the value 0.994 or whatever, right? Which is actually the result of the tan h of x, right? And now I can compute e to the 0.99 whatever, and I don't know exactly what that is, but it should be something like 2 point, it's basically still e to the 1, so 2.71 maybe, something like that, ... right? And so now answer takes on this value 2.71. And the next step, if we go up here, is that answer is assigned equal to the square root of answer. And of course this is completely legitimate, because when R looks that up, it's going to find the value 2.71 here, right? Compute the square root, which I don't know what that is probably something like 1.2, I don't know, right? And then it basically rewrites this now to be 1.2 or whatever that value is, the square root of 2.71. Okay, so you know, to go back to our example here, there's maybe slightly more eloquent, because we don't have all of these extra little variables hanging around in our environment at the end of the day. I mean, if somebody was to be picky, they'd say, well, you create this variable x1, x2, x3, but you never use them anymore, so they're just hanging around in your environment. If you go to your environment in RStudio Cloud, you'll see all those variables are created, and you'd have to manually delete them. So maybe this is slightly more eloquent. Okay, so level 3 views these things as composition of functions. Okay, so here our observation is that x is successively transformed by different functions. And so the tan h is the first level, the deepest level, right? It's the first function. And the result of this function becomes the input for the exp function. And the result of that becomes the input to the square root function. So the functions are composed from each other, and lastly for log 2. Now, that's a perfectly legitimate, nice, elegant solution. We don't create any intermediate variables, and at least this example, it's quite readable. However, that's not always the case. in R. Sometimes we have a long chain of compositions, far more than 4, and some of those compositions have a lot of parameters, or um, uh, things have to happen in between, etc. It gets quite confusing to write in this notation. So what pipes do, and that's approach 4 here, is um, essentially like change our perception of X to be something that's acted upon through a like kind of a pipeline, right? A sequence of steps. Sorry, my dog wants out again. So here we start with X, and we say X is piped into the tanh function. So the Magritta package is smart enough to know that this X becomes the first parameter for that function tanh. So it's equivalent to tanh with X. We don't need to explicitly say here that, it just knows. And then the result of that, right, that's the innermost function, is then piped into the exponent function, which in turn is piped into the square root of function, which is in turn, etc. Now, this is interesting. Note here that I use this symbol that we haven't seen before. It is perfectly legitimate to write X is assigned equal to 5, but you can also in R write 5 is assigned equal, whoops, oh my, okay, I'm sorry, 5 is assigned equal to X. You just have to reverse the direction of the assignment. So dash greater than is assigning it in this direction, the direction of the arrow, obviously. Okay, so the left-hand side is piped in to the first argument of the function, right, appearing on the right-hand side. If it's not the first argument, you can use the period as a placeholder. So for example, we saw before this example of the seek function. So here I'm piping in the value 10 into this parameter here, but I want, remember this was this to and from, so the first one is from, this is the from, and this period here is going to be replaced by the 10. Okay, so this 10 is going to go in here, and this is the by equal to 2. And so the result of that first guy here is 1, 3, 5, 7, 9. If I pipe 3 into this function, but now I've used the period down here, okay, that means it's going to replace there. That's the 3 is going to go in here. Then it's just essentially saying count from 1 to 10 by 3, so I get 1, 4, 7, 10. Okay. If I do this, 5 is piped into this call. Remember, if I name my parameters from 1 by equal to 1, what we're missing is the 2. From 1, 2, and that's going to, R is smart enough to know to replace 5 by 2 is equal to 5, and that's in fact what you get. Okay. All right. An interesting point here that we haven't dealt with in the course is, why do we use this assignment or this assigns to throughout our code, but then in function calls we use this equal? The reality is that actually a single equal is equivalent for all intents and purposes to these guys. The issue though is that we know that there's this double equal, which isn't assigned to. It's like testing if two variables are equal, they have the same value. So I would prefer that you use this, but in function calls, the standard in R is to use this equal sign. Okay, we can return to our example from last class, lecture 5 at the end. Recall we were looking at dplyr, and we applied a fancy set of dplyr functions to our data. If we want to write that as a pipe, no problem. Here I see my starting data set is here, so it goes into rename. I just use small bracket, and it's piped into the rename function. Notice here there is I don't need to explicitly say small bracket here, like I did up here, because it knows that it takes the first argument. And then this part here is exactly the same as this part here. Nothing changes, okay? Now the results of that are piped into this filter function. Now again, I don't need to write B0 here or anything because it just takes the result from rename, okay? And the rest of this is exactly the same as here. And the results of that then, I think that it's scrolled off the screen here, but then I pipe that into this select, right? And again, I don't need to state anything explicitly because it's assumed that the result of this filter will go in here at the beginning, right? And that in turn will go into this filter with vital state is dead, same as before, which gets piped into mutate, same mutate as before, and they're exactly the same things. They do exactly the same things. Now, I think here, you know, it's not really clear perhaps what the advantage of pipes is. You know, is it more or less readable? I don't know. I mean, it's a bit of a esoteric thing at this point. But you'll have to trust me that later on in some examples it gets much nicer when you use pipes. Okay, so let's return to the breast cancer transcriptome now. Okay, last week we learned how to winnow our data set down to the core, and we've seen how to visualize that data with ggplot. Now we want to focus on the types of questions that we can ask, and how to model our data, and how that in turn then allows us to refine our data selection and visualization. And as I mentioned at the beginning of class, modeling is a huge topic. Which model you use depends very much on what your problem at hand is. So we can't cover all that today, but we can try to get set up. Okay, some fundamental concepts really. Let's see if I can get these across in a pseudo-elegant way. Look, domain-specific knowledge is really important, okay? It's true in data science and all areas of science, and it's going to stay that way for a long time until we have some extremely fancy artificial intelligence. You know, we need to listen to the experts to tell us what the individual variables mean, and also just why track those variables in the first place. I mean, presumably they track them because they are predictive of, for example, you know, a clinical endpoint like patient benefit from therapy. Okay, so they measure, let's say, the protein levels of a small number of targets like the estrogen receptor and HER2. They keep information like staging, and they keep information like lymph node status around because they think that they will tell. They will give some information that will suggest whether the patient will or will not respond to tamoxifen. Okay. Now, that's perfectly valid, but there's a part of data science which is also which aims to stand back and be very objective and let the data speak for itself. Now, in our data, we've also done gene expression profiling. So we've used RNA sequencing to estimate the counts of every gene, right? Not just estrogen receptor and HER2, but every gene in the human genome. And we want to know if any of those genes, there's 20,000 or so, any of them inform as to whether the patient will benefit from tamoxifen. So we're doing discovery science. And we don't want to restrict ourselves to just what the experts have been doing, right? I mean, the experts themselves are the first to say that those classic techniques are not optimal. And that's research, right? Okay. So we know to death now that the columns in tables correspond to variables. Here, I just want to say that variables can take on different values. Now, this is not the same thing as the type of a variable. It's a bit different discussion. The first, some variables are categorical, meaning they can have a small range of values. For example, x could have the value, the integer value 0, 1, or 2. So, you know, it's of class or type numeric or type integer, but it's restricted within that domain to just a small range of values. A tumor could have only values true or false. For example, salsa could have values hot, medium, and cold. So there's really these distinct, discrete levels or values that the variable can take. In some cases, categorical variables are ordered. For example, the days of the work week at least are ordered. Sunday to Monday, Monday to Tuesday to Saturday. We think of them as going as progressing linearly through a week, even though the days of the week are ultimately cyclic because you go from Saturday back to Sunday. Stage variable is another example. We have stage 1 and 1A, stage 2, etc. On the other hand, some variables are continuous, meaning they have an infinite number of values, and they're ordered. For example, temperature is a real number, right? You can have 21 degrees, or you could have 21.01 or 21.000001, etc. So there's an infinite number of values there. And the expression of ESR1 is a continuous variable, or is it? Well, I'd like you to think about that. Maybe stop the lecture for a minute, or think about it afterwards. But while we're counting the number of our expression measurements of ESR1 are counts, transcripts, so that's kind of an integer. I don't think you can have infinite number of copies of ESR1. So, yeah, maybe you could model it, but it's a bit tricky. I think it's probably wise to model it as a continuous variable. So categorical, when you talk about visualization, you usually think of bar plots when you talk about categorical, and histograms for continuous. So let's look at that a little bit now. Without further ado, we return to our studio in R. There's a Lecture 6 project available there, and it has this code and data. I'm still using the small bracket data set, but for convenience here, first I'm going to transform it in two ways that we did last class. I'll get rid of this AGCC long stage variable and replace it with just simple stage. And I don't need some of these different variables, so to simplify it, printing out, etc. I've selected against them with the, see that, the negative operator, right? Removes those columns. Okay, and then I jump straight to ggplot. I tell it which tibble to use, which is BRCA, and then I set my mapping, which this is a bar plot, so you only need to specify the x-axis. So here my x-axis is stage, so stage 1, stage 1A. Notice I haven't gotten rid of those weird guys like I did last class. And something's gone on here because actually ggplot has computed then how many of the samples of the 1200 samples belong to each one of these classes, right? Okay, and you can see really that what we saw last time when we filtered out the weird stage guys, that there's not too many of those abnormal guys. That majority of them in our data set are really stage 2A, it's a huge class, and stage 2B, and there's fewer stage 4, etc. Okay, so yeah, now the bar plot, yeah, so you only, in a bar plot, you only need to actually specify your x, and ggplot computes the y as just a number of rows that fit into each of the values that stage can take, right? And that's possible because stage is a categorical variable, and so there's only a small number of different bins to put those patients into, or yeah, patient samples. Here I'm just throwing in some little examples here of how you can fancy up your graphs. So change the angle to 45 degrees to make it more readable, and adjust it a little bit horizontally, how to shift it, etc. They're just small tips that you'll pick up as you go along. We can look at other variables in the same way with a bar plot. We could look at least all the other categorical variables. So this gives us the variability of that variable, right? What values does it take in its space of values? Stage 1, stage 2, stage 4, etc. NA. What values does it tend to take on? Um, Now, so I can put on several bar plots on the same um, picture, same figure. I'm going to use this nice thing called package called ggpubr. So I have to load that library. I've already installed the package on RStudio Cloud, so you have access to it. And then there's a function called ggarrange that you'll see allows me to put different bar plots together. So I'm doing the same thing, but for gender. Um, this is our gender thing. So my x-axis is gender for menopause status, race, and ethnicity. Okay, so it's 4 different bar plots, and now I can combine them using this ggarrange function where I just pass it in the 4 different bar plots, and I say, okay, I want 2 columns and 2 rows, and I get back this nice figure here that looks like this. Okay, so now before we get into the interpretation of that, I just want to point out here that I can actually save a ggplot as a variable. So this is one ggplot, one bar plot, and I can save it back, assign it to a variable called ggender. That's an arbitrary name I made up. Graph gender, graph menopause status. So if I want to render that plot on the screen, then I just need to actually type ggender, and it'll show up on the screen. But here, in this case here, I pass plots to a function, right? So they're now arguments to a function, and this garrange just put them together nicely like this. And actually, there's some interesting findings here. I think I didn't know there's actually is a male, at least one male sample in our data set. That's the source of variability, I'm sure. That's interesting. I haven't looked at it explicitly. So there's something where I would keep my eye open for. So I would probably go and get that participant's code and try to memorize it or mark it down, and then be sensitive in the analysis to see if that's showing up often, because it could really be an outlier. I mean, there's a lot of differences between men and women at the molecular level. And I would think that male breast cancers are, well, they are actually not that different from female breast cancers. We know that now. Some really famous researcher in Holland, she has really been kind of focusing on that for years now, and they've looked at hundreds of male breast cancer cases, and find that they're actually much like some of the luminal breast cancer cases in women. Okay, so if we go over to race, now you can see that my wrangling of the data into RStudio wasn't so great. So these are really long labels, and they kind of screw up the plot. In fact, like they cause it to be smaller, etc. I would probably go in and change those different values for that variable called race to make it a bit more convenient. But you can see that most of our data set is white. I would say that's probably about 900 of the 1200 seem to be white, with some African American and Asian. Our gender, ethnicity, it's not Hispanic or Latino, so I guess that means that it would be, I don't, we'd have to actually ask what ethnicity is exactly, but it's just basically, I guess, anything other than Latino. I'm not sure why they've coded it this way, but the vast majority is, of your data set is not Hispanic or Latino. Race, menopause status. Most of the women in our set are postmenopausal. You can see that here. A few, I would say, what is that, about a little bit more than a sixth of our data set, they are premenopausal, and then there are some special cases like peri, where they're in between, I guess. I'd have to look up the exact definition. Okay, so we can look at some more categorical variables. Histology, that's what the tumor looks like under the microscope. You can see the figures have been completely destroyed, at least partly because there's so many different values that variable can take. It's still categorical, but it's not really a small number of categories. there's lots of different ways to classify a tumor, and it's so many, and some of them are very long names. So they've messed up the formatting of the figure. We're going to return to it on the next slide. Age also, in principle, has many different values, but here in TCGA, this was just the year, not the number of days since birth. It was just an integer, like 61 years of age or 75. And I could have been a little bit fancier. They're squished together here. If my screen was bigger, I could widen it. But you can see at least that there's a good amount of variability in the age of the patients. Okay, some of these, this is NA down here. You can't see it too well. But we do see clearly a lot of variability. Recall there's 1,200 patients in total, and only this guy here is like 58 or so patients of that. So it's really quite a nice broad distribution. Micromets detection by IHC. So what is that? 300 of 1,200. 1/4 of our data set have had, they've detected micrometastases in the patient post-treatment, okay, which is not good news at all, right? Lymph nodes examined count. That's also quite interesting. We have many that we don't know, not available, and there's also this NA encoding down here. But we saw this in a previous slide a couple weeks ago. Again, I could fix these labels a little bit more. You can make them smaller or bigger, but they're also not ordered very well here. But you get the basic idea that there is quite a bit of variability with across the lymph node spectrum. Let's dig in a little bit into this histological diagnosis. That was the figure in the top left that didn't look very good at all. So what I've done is use the group by and summarize functions in dplyr to get an idea of what's going on. So here I pipe BRCA into group by. Now remember that piping conceptually it puts BRCA into here, so we don't need to do it ourselves. So BRCA is piped into the group by. Now group by reorganizes our data by the different values that histological diagnosis can take. Okay, you'll see here that we actually have 60 possible values for histological diagnosis. NA, adenocarcinoma with squamous differentiation, metaplastic, adenoid cystic carcinoma, apocrine, etc. Okay, now I also included here, I then piped the result of that grouping by histological diagnosis, putting all the patients with the same histology together. I pipe it into the summarize function, which counts how many variables in each category. That was very strange. Did you see that? Like a pizza. I don't know how I got that thing. Very interesting. I learned something new about our markdown every day. Well, what you see here is that the vast majority of our samples, what is that, 1,100 out of 1,200, have not applicable, and then there's like 1 or 2 for the remaining guys. So, I'm not really sure the value of this variable in our data set. So few of the patients have the same histology, and the vast majority they don't even have a histology for. So that would be probably a variable I would think about maybe ignoring in my analysis.
Okay, this is the end of our categorical variables, but I need to make a mention of something that might be confusing to people. Our data set actually has 2 measurements of the estrogen receptor, progesterone receptor, and human HER2 or ERB2, as it's officially known. Now, these measurements here are from clinical or in the pathology from hospitals that use IHC for ER and PR, okay? And this measurement here is coming from a different kind of assay, but also a clinical assay called FISH, okay, fluorescence in situ hybridization. Now, these aren't gene expression measurements. Now, all three of these proteins have genes in gene expression side of things, and they're called ESR1, ERB2. I think the other one is PGR for the progesterone receptor, and for HER2, it's this ERBB2. Okay, so these guys here, these variables in the gene expression side are exactly that. They're estimates of the transcriptional level, where these are estimates of the protein level, and for the fish, it's measures of the DNA copy number. So actually, it's something interesting that we can compare. And I think if we look closely here, we see already some interesting things. Most of our data set is ER positive patients. That's consistent with the literature and epidemiology of the disease. And we only have about, I think that I calculated this was to be about 10% of patients to be, 10% of patients are about, or even less, are HER2 positive. That's a bit lower than what we'd expect in the population. Okay.
Okay, but we really want to explore the new data. We want to explore our gene expression profiling. But we would note first that that's continuous in nature, that the counts and the estimates are large numbers. There's no small number of bins that the genes take. So there need special ways of visualizing continuous data as compared to the categorical variables we've already seen. And one thing we would use is the histogram. Okay, so that's a classic visualization technique. So a histogram partitions the x-axis into equal-sized bins across the range of the target value. Okay, so let me draw something out here. So let's suppose I had some data, and this is my x-axis, okay? And it goes on in both directions, but my data only ranges between -5, that's the minimum value in my data set, and the other side, my maximum value is +15, okay? Max, alrighty. So now I have patients, samples that have ESR1 values that fall between this value, and I can choose the number of bins, okay? Or I can choose the bin size. So let's start with bin size. And let's, maybe as a good first choice, maybe we'll make that into 5. So that means there'll be a bin here from -5 to 0, from 0 to 5, and from 5 to 10. Okay? So we could label this bin 1, bin 2, bin 3, bin 4. Okay? So that's the x-axis. Now, Spart, that's my dog. That was the x-axis, and now the y-axis. The y-axis then tabulates how many patients fall into each of these bins. So for example, if I come back here, my y-axis would go up like this, okay? And on my y-axis I might have 1, 2, 3, 4. And my data, I might observe that there's 1 patient here in bin 1, no patients in bin 2, 3 patients in bin 4, bin 3, and 4 patients in bin 4. And that's a histogram with bin size equal to 5. Okay. So what I try to introduce here is some new notation that's kind of handy as you're exploring R. This is the cat function. So the cat function is basically a way of sending characters to the screen in a nice, tidy way. So here I create a character that's the min value, and then I have a comma that separates it, and the min function takes what you would expect is the minimum, and I use here BRCA $ESR1. So recall that the dollar sign gives you access to that variable, just that variable, that long column of 1200 ESR1 measurements. So I'm asking for the minimum of all bracket ESR1 measurements, max value, and then the max function, which functions obviously analogous to the min function. And then, oops, I come in and I ask for the number of observations, which I can use the nRow function to look at. That gives me the number of rows in the bracket tibble. And finally, I have one more string with this weird symbols in here of \n. So they require maybe some discussion. So the \n is new line. It's historical. Whoops. It's historically how we write or tell the computer to print a new line to the screen. So to take the cursor and move it to the beginning of the next line. And so here I've asked for 2 new lines in a row. Finally, so yeah, I put one at the beginning too, so that it would start out. And you can see here now the result of that cat function is min value 16, max value quite large, almost 200,000, and the number of observations is 1215. The last thing I did over here was ask for bracket, again, $ ESR1, but here I've asked just, it's a vector now, it's just a long column of numbers for each patient, right? So it's just one long column of 1200. Here I've just asked to write out the first 50 entries in that long vector. Okay, and so what I see then, sorry, I want to shut that off. What I see then is that the first 50 values here are printed out, value 1, and then this would be value 50 down here. You can see that there's a huge range in the values of ESR1. In some cases here, it's as low as 83, and in other cases, the maximum, for example, was almost 200, whereas that guy, I guess it's not in the top 50, but there's no shortage of guys that are quite big. That's 133,000, 129,000. Okay, we're going to come back to this, but when you see data that ranges over so many orders of magnitude, from 10 to the 1, like 83, maybe it's 10 to the 2, but still, up to 10 to the 5, which is 200,000, you know, that suggests maybe you're going to need to transform the data. Often, gene expression data is log transformed, and you're going to see that we do that later on. This is a histogram, or a series of histograms for ESR1 gene expression. In the first one here, it's by default number of bins, which I believe is 30, spanning this region from 0 to 200,000. It already tells a story, so it says there's quite a few values in the low range, and a lot of outliers here at these very high ranges. And we can change the bins, the number of bins used, to 100, so about 3 times that. And I think the story is refined a little bit to show you that many, actually, let's say 250 of the 1200 measurements there in total are very close to 0. So now in the other direction, if I decrease the number of bins and I lower it to something like 10, well, you can see the effect that it looks kind of almost cartoonish in a sense. And the last plot here is also a histogram, but I'm just showing here that you can change the limits on the y-axis. You can always change the limits on the x-axis too, and there's much more functionality that I don't show here. Okay, so just as a semantic ARP thing here, ggplot thing is, notice I create one variable g that holds the generic, let's say generic ggplot for on the tibble for ESR1, and then I modify that same g in different ways, g1 through g4, and pass those to my GG arrange to get this nice representation. Okay. So histograms have some limitations. The first of which may be that the choice of the number of bins, which determines how wide each bin is, can greatly affect how you interpret the plot. So for example, back here, if I was to only look at this very coarse, large bin width, I wouldn't see that many of my points really are focused on 0, which I know because I have actually explicitly looked at this more refined perspective. Yeah, so I think you kind of visually see how things do change a little bit, and I think your statements might be slightly different depending on which ones you looked at. And the second thing is that the fact that there is a large range of values and these like big outliers make part of the histogram to be somewhat, let's say, vacuous or empty, and that comes at the cost of not being able to see important structure in other parts of the distribution. Okay, so for example, here there's just a few points above 100,000 really, yet they take up half of this plot, and this half of the plot is trying to express what's going on in the 1,100 remaining points. So a small fraction of the values dominates the landscape. We would like to maybe have better resolution in this area here. Right? So histograms are not very robust to those outliers. So now, what are outliers, right? That's a really important question. And it comes down to, and how do you deal with outliers? It somewhat comes down to what the source of that outlier is. Sometimes there's a particular observation for a sample, in this case here, a patient, where the profiling technology didn't really work very well. And so all the genes for that sample have aberrant outlier behavior, or many of them do. And if that's because of a technical problem, it might be justified to remove that sample from the study. Or perhaps if it's just one gene, like ESR1, that's an outlier in expression, then, well, it could be that maybe it was entered wrong, or there's been some kind of computer error in recording of that value. That includes, say, the code, like our code, well, not our code, but somebody else's code that has a bug in it that incorrectly processes the information. So in these kinds of cases, either you can fix the bug, or you can throw out the sample if it's really hasn't worked. In other cases, it may represent true biological variability, and it's an art more than a science of trying to figure out which it is. If it's true biological variability, it probably shouldn't be thrown out. Now, if your question is about visualization, at times with a histogram you might remove the point just so that you can go back and look at those areas of the graph you're more interested in. For example, if you were to remove all the points above 100,000 here, you would then use this territory to zoom in onto this region. So that's also valid to do. Is you're not throwing out those points from the study, you're just throwing them out from the figure so that you can narrow down your or you can investigate more closely. Okay, so how do you hunt for outliers? Well, we have all those tools from the previous class, and for example here I've assigned as a result to expression the piping action here. So I take bracket, I pipe it into my arrange function, and here I'm going to use a new function we haven't seen before called descending. So if I had removed this descending, it would order an ascending value by count, ESR1, so from left to right in that histogram, but really I want right to left. Those guys are the right tail, those large values, and so I can use the descending to invert things. So after arranging by ESR1 levels, I can select all those variables from participant to tumor status with ESR1. So I'm throwing out all the other genes here just for purposes of exposition. and I print out the resultant Tibble expression, or the first 15 lines of it, and I can see that the value starts at 198,790, and there's two of them from two different patients. One's white, one's Asian, so they're definitely not the same patient. Then there's a bunch at 16, 7, 8, 8, 9, and then a few at this level, all from different patients, and they do have some variability in their other variables. So they're not all Asian, they're not all menopause post, they're not all not Hispanic, in fact. So it's hard to say that there's a certain kind of patient that's causing these very high values. It is suspicious to me that there are repeated numbers here. Now, I think that might come from a normalization procedure of the data that was done by TCGA. It's a bit strange to see such counts that are exactly the same, and I would have to investigate that further. I didn't know that they existed until I made this plot. In fact, it's actually very interesting, and from my perspective, not yours. Okay, so what we really need to do is look at ESR1 expression of these outliers against the expression patterns of the other genes, right? And that's going to give us a little bit of a frame of reference for whether 200,000 is really an outlier count or not. Okay, so now one of the approaches that is the box plot. Now it's already the dinosaur visualization, I would say. We're going to get to replacements in a minute, but it addresses actually both complaints of histograms, number 1 and 2, in terms of the effect of outliers and the choice of bin size, etc. So a box plot, and these are not great examples here, but because they're so squished, because this plot really needs a log transform. But I'm going to first show you the syntax in ggplot, and then we'll get to some nicer examples of box plots to show what they do. So here I'm going to take my BRCA tibble. I'm going to pipe it into a select to just pick a few genes expression, just to narrow it down to 5 or 6 or I guess 5 for visualization purposes. So just using those 5 genes, I'm going to pipe that into a function we haven't seen before called MELT. Now I'm going to show you a whole slide about MELT in a second, but right now this is the summary here is I'm going to change this BRCA structure, or the reduced selected BRCA structure, into and I'm going to rearrange it a little bit so that each gene is an observation. I'll show you in a second exactly what that is. And then we get to the ggplot finally, and that ggplot takes this lava, that's the output, the melted tibble. The melted BRCA tibble is now a lava tibble, and that's sent to ggplot. Here my variable is going to be one of ESR1, PGR, ERB2, EGFR, or keratin 5. So this MELT function, it created from what are called the measure variables here 5 new variables, or sorry, yeah, a new variable called variable, not, it's an automatic name. And along the y-axis, this axis here, is the value. So that's the count for ESR1 and PGR, ERB2, EGFR, and keratin 5. Okay, so this is not the nicest box plots to express exactly what it's trying to depict. And that's because this data really needs a log transform before we continue. So I'm going to do that in a second. Okay, so I'm going to begin by describing the MELT function that's in the so-called Reshape 2 package. This is a popular package. I've already installed it on RStudio Cloud for you. It's a bit confusing at first, but there's an assignment question that asks you to explore it on your own, so it'll help you get it straight in your head. It's an important function. Okay, so we start by this observation again that in our tibble currently, the small bracket, the value for all 50 genes are contained in the same row. Okay, so in other words, if this is our tibble for one patient here, let's call it patient P, well, we have something like 79 different rows, and the last 50 of which, these guys, that's the 50 genes, right? And so what I mean is that the expression level of aniline, ESR1, ERB2, and TM-EM45B, they're all in the same row here for an individual patient P. ggplot doesn't really like that, many of its functions. They want the gene count for each gene to be on a separate row. Okay, so I think an example makes this much more clear than words can. So suppose I create a tibble called Timmy, okay, and in my Timmy tibble, I have just 2 patients, not 1,200, just 2. And I only have 2 genes, ESR1 and ERB2, all right? Okay, so it's a minimal example to show how this works. When we melt the Timmy tibble, we're going to have a new tibble that is has 4 observations instead of 2. 2 of the observations are for patient A1NF, and 2 of the observations are for A27. In the first patient, A1NF, there's 1 entry, 1 row for ESR1, and it's going to be a 5 because it's 5 in this matrix. And the second row is the entry for ERB2, and it's 100 because it's 100 here. Same thing for A27. It has an ESR1 observation and an ERB2 observation, and they're going to be 50 and 10 respectively. Okay, so maybe you want to pause the lecture right here and ask, is all the information in the first original Timmy tibble also present in the melted Timmy tibble? Okay, that's the first thing. And then if you're more energetic right now, or you just want to break from listening to me moan on, then make a small example that contains 3 variables, and let's say 3 patients if you're really ambitious, and show what the melted new Timmy tibble would look like. Okay. So before showing you how to melt bracket to lava, this is the code for melting Timmy to as in the previous slide. So there, I would say I want to, my ID variable is the participant, that's A1NF or A27, or 827, what was the patient name? It's the it's this first column here, the participant. That one is, we want in some sense to stay the same, and that's the ID. And the second one is called the measure variable, and we want to do that. We want to separate ESR1 and ERB2 onto separate lines, and that melt there would of course go from Timmy tibble to melted Timmy here. Okay, now here's a larger example with the BRCA data set, where I first select, let's say, a few genes, just 5, the same 5 as I used before, along with the participant in tumor, and then I'll melt them. My ID vars are going to be participant in tumor. They stay on there as before, but the variables that we want to have on each individual line would be these guys, all the genes from ESR1 to keratin 5. And if I take a look at the resultant, okay, this gets assigned to a new tibble called lava here. And if I look at lava down here, I can see that the A1NF, A27, AOGZ, etc., all of the 1,200 or so participants, they all have an ESR1 value here, and that would be, and this is the value for those ESR1 counts. Okay, so now you'll notice that for every sample A1NF, it's going to have a row for each gene. So if we had, in our example here, 5 genes, A1NF is going to show up in 5 different rows here, once for each of those genes. And so if you have 1,200 samples in total, and they all are now multiplied by 5, that's why you get something like 6,075 total rows. You can validate the arithmetic on your own. Okay, I hope that's understandable. So the other thing here that I go back is that because we have such a large range of magnitude in the counts, we really should do a log transform. And now we can talk about the power of the box plot. So first is, how do we do a log transform on this data? So now with the lava data structure that's been melted, our variables on the x-axis are our 5 chosen genes. The value on the y-axis is the value. We're using a box plot, so we set that in the geome box plot. Our scale here on the y-axis, so it's the counts, is log transformed, and that's how you specify that in ggplot. And this last theme here is just for making the labels a little bit prettier. But you can see here now you have in a box plot this center region, okay? And in that center box, this is the box of the box plot, this line in the middle, that's the median of the distribution, okay? So, well, you can see that the median across the 5 genes does change quite a bit between the different genes. This upper line here represents the 75th percentile of the distribution, and this line represents the 25th percentile. The whiskers, this guy here and that guy there, is 1.58 times the this region here. Basically, if you go from the end of this whisker to that whisker, it's a region that contains about 95% of all of the points. Okay. Now the last thing is that you see these dots here and here. These are individual points that exceed the 95% confidence interval. Okay, so I think looking just at this distribution now, this kind of nice box plot, okay, I should say, let me just skip back. You can see that information that I just spoke about here, that I talked about. I just didn't want to talk about it there because it's a bit noisy. Okay, I think that actually there's already some interpretation here that we can make. Notice that there's a lot of outliers for ERB2. All the outliers for ESR1 are at the bottom end. So there's a few patients that have extremely low ESR1 expression. The medians, as I said before, seem to move quite a bit. Like the 95% confidence region of PGR, from here to here, well, it overlaps with all of these guys, but you know, the 25 to 75% confidence interval of ERB2 is higher than even the 75th percent confidence line in PGR. So I'd say there's a pretty good amount of variability. And the other conclusion there is that I don't think that if we look back now and say those counts that we observed for the outliers in ESR1 are so high that they should be removed. That's these guys up top here, and there's actually almost, I guess, every gene has values at least as high as that. Okay, well, box plots are box plots, but violin plots are way cooler, and I give a +1 to anyone in the course to tell me why one would ever use a box plot instead of a violin plot. So, yeah, you can see here some example of violin plots. The bar here indicates the median as before. Now you can see the actual probability distribution function along the edges, so it's symmetric in this case, across the, this axis here. So you know a little bit better than a box plot where your points really lie. Okay, so it gets this bulge here means that the majority of your points for aniline lie here, and it gets narrower and narrower as you go towards the tips. If you look at, for example, in the normals for FOXC1, this distribution here, it's very interesting how, this is not the normals for FOXC1, it's the normals for CDH3. So it's normal because tumor is false. Okay, look how far down it dips. Isn't that an interesting distribution? Yet overall, its median is higher in normal tissue than in tumor. Okay, so I like the violin plots a lot, and to do those in R is quite simple. I had to melt, as before, the BRCA tibble into lava tibble, and then my GG plot plots my variable versus the value after the melt statement. And my fill here is tumor, so that's why it's been split into tumor and normal. I just wanted to compare those two. Geome violin is the right function in ggplot. And then basically you can ignore these little conditions here. They're just to make it look a little bit nicer, okay? I've log scaled the y-axis this way again. Okay. Well, I think already you can see some very interesting differences between tumor material and normal. Almost, many of these genes, there seems to be quite a significant difference. Now, significance should be, there is, statistics would be where you would turn to evaluate whether there's a formal difference between these two distributions, and that's very, very important. But I think that we can see visually that aniline, for example, UBE2T is very different. NDC80 looks almost identical and different. ORC6 is also quite different. ESR1 here, that's one of our famous guys, of course. I don't see very much of a difference in terms of the median, but the distribution in tumor is much more varied. It has both more extreme in the positive and negative directions. Okay. So just for fun, I plotted all 50 genes, tumor versus normal, in our data set. And I think this is actually extremely interesting. For example here, the keratins, which are markers of epithelial cells, which are often lost in breast cancers as cells lose their adhesion and start to migrate, has a huge variance in the tumor samples compared to the normal samples. And that kind of makes sense. For another example here, let's take MDM2. They have it has very tight expression, both in tumor and normal, around its median. So the whiskers here are quite short, meaning that patients don't vary much different between tumor and normal. EGFR is a big player in triple negative breast cancers. That's those cancers that are both ER and PR and HER2 negative. Maybe another example here is UB2T, and you can see, if you look closely, that the expression in normal is far lower on average than in medians. And aniline also has that behavior. And the third one here is ORC6 that I picked out after staring at this for a little while. Now, where is ORC6? Oh, sorry, it's painful to... Oh man, I have lost ORC6. There it is, right here. ORC6 also has a pretty good difference between tumor and normal. Okay. So I find already that this figure is quite interesting to stare at and generates lots of hypotheses, although it's admittedly far too noisy to be ultimately very useful. Okay, so, well, we could ask then, could we model the tissue status, tumor versus normal, using gene expression? So in other words, could we just look at the expression levels of, say, aniline and UBE2T to differentiate between healthy and breast tumor material? There could be clinical utility in that, but probably this is a bit artificial as a class exercise. But, well, I think it's an interesting place to start. So what if we just plotted our tibble, BRCA? X is the expression of aniline, Y is UBT2. Notice I've now scaled both the Y and X on a log scale, a log transform, okay? What else do I have to say about this? I color the tumor status by blue and orange. So blue here is tumor, and the orange are quite low. And I've changed the size of my points to 0.3, as we saw very early on in the course. I added one thing here that's I think is kind of cool, is this genome rug, which basically plots the points, projects the points onto the y-axis and onto the x-axis to get a feel where the density of the points lie. I don't know, I think it looks kind of cool. Sometimes I find it useful. To me, it seems very clear that if I was to use either of these genes in isolation, I would be pretty good at predicting whether it's normal or tumor. So, for example, maybe I could draw a magic line in the sand right here, because I would say below this point it's mostly red, and above this point it's mostly, or maybe orange, I guess that is, and this is green or turquoise. Now, there's some mistakes like here, but I think that's a pretty good line to call it. So if I drew when I was to go and extend this line over here, any point above this line I would call tumor, and any point below this line I would call normal. Now it's going to make mistakes here, all these orange points above that line, and all these blue points below that line I would call, well, falsely call normal samples, although they're tumor. Now I could also look at aniline and say, well, if I drew a magic line here, then all those lines on this side I would call normal, and a lot of times on all the points on this side of the line I would call tumor. And again, I would make mistakes. This guy here is a normal sampler, but I would predict it to be tumor based on just aniline's expression. So the real power here is when we use both aniline and UBE2T together to classify whether the sample is tumor or normal. And this is going to come up an awful lot in the course. So how do we combine multiple pieces of information together to make predictions, or to make classifications, to, in this case here, differentiate between healthy and breast and tumor material? Okay, and that's going to come up in many different contexts. So let me show you a little bit more where this is going. And now we're getting into the modeling part of the whole data science cycle. Okay, so the last couple slides here are just for your interest sake at this point, to give you context of where we're going. What I'm going to show you now, I'm not going to explain in depth, and it might be very difficult to understand, so I don't want you to panic here. Okay, so here's the idea that we're going to see often in the course. We're going to try to use the gene expression data to predict or classify a property of the tumor or the patient. So let's suppose we wanted to, here's the scenario. It's a bit artificial, but it's a good place to start. So imagine a woman comes into the clinic and there's reason to believe that she has breast cancer, and so a biopsy is taken, and we profile with RNA sequencing the transcripts in her breast material. Could we use aniline and UBT2T, their expression levels, to predict or determine whether her tissue is has a tumor or not? Now, this is a very artificial example. This would never work,
because a biopsy is very local. It wouldn't tell you where in the breast. I don't think that this is really, but it's a good example maybe. So this, we would create a model, okay? And in R, we're going to get to this. There's a nice package called Model R that has functions like GLM and many others. This stands for Generalized Linear Model. I'll foreshadow here and say that we're going to look a lot at something called logistic regression later on, because it's also the basis for deep learning and it's used. It's a classic statistical method. Now, the point I want to really kind of make here briefly is that we're going to create a model that predicts tumor type. Remember, in our Tibble, tumor was true if the sample was actually tumors material, and false if it was morphologically adjacent normal. No, right. Okay, so normal samples. So we wanted to predict true or false for a sample based on the expression of aniline and the expression of UBT2T. Now, logistic regression is one way that we could explore that within our bracket Tibble. Okay. Now, when we build such a model, we can query R with the summary function for how good that model is. And we're going to go into this in more detail throughout the course. But it turns out that actually the short story here is that we can predict the status of a tissue, if we don't know it, into tumor versus normal with extremely high accuracy based on just the expression of aniline and UBE2T. Now, I don't understand, again, I also don't really know here whether this makes any sense. Should aniline and UBT2T as genes in their role in the cell be able to predict or distinguish between tumor and normal? That's another good question. Some points of reflection for you that I think you could consider, and we'll speak about these in the lecture. So that's the end of the first part of the course. I hope you're hanging in there. By the end of the course, trust me, these things are going to be second nature to you, so there's no reason to panic. Thank you very much, and I'll see you again in the lecture.